Thank you, Professor Bell. Hello, everybody. It's kind of a minimal public, but it's still, I hope, interesting matter. Therefore, I will try to introduce within the next hour protein structure and how this is related to cryo electron microscopy. If you have any questions about the theory and application, please do not hesitate to email me. If you have any project you would like to run at CNS with our Cryo-EM setup, please do contact me at the email below. All right. Oh, why is not doing that?
You have to have
okay. I have to use the arrows. I cannot use the arrows on my keyboard. Okay, then my first slide is about protein. Just a short introduction in case you haven't got this class in your early studies. And it's just to clarify the terminology, because proteins are referred in different way depending if you come from physics, chemistry, or biology. Then generally, proteins are considered as biological macromolecules or biomolecules, and they differ from each other by their amino acid sequence. This amino acid sequence is defined by the nucleotide sequence in our genes. And from this point of view, protein structure is the last step in the central dogma of molecular biology, which means that the information flow of basically how life is constructed, the life we have, is that we start from the DNA information is transcribed into the RNA information is translated into protein, but this is not sufficient for us to function. Protein has to be to have a structure, and then this structure translates in the last step the protein function. There are 20 unique amino acids which consist a protein, and from this point of view, protein can be also defined as a polypeptide chain. Polypeptide chains officially are considered when there are more than 50 amino acids, but this is not sufficient. A polypeptide chain is not sufficient to make a protein. These polypeptide chains has to have a specific structural organization. The tertiary organization of these chains define the protein structure, and the quaternary define its function. Cryo-EM, from the perspective of protein structure, has evolved in this method of excellence to define the quaternary structure of proteins, and thus give us a direct visualization of their function if we have the tool and the knowledge to translate this information from structure to function, which is the last step of the central dogma in biology. There are more than 20,000 proteins encoded by the human genome, obviously much less in other species. Now, I quite like this graph, and you have all the additional information in the link before it, because it illustrates exactly how the information flows in the cell. And what is interesting to know, that the step of protein translation is actually happens in different compartments of the cells. It gets out of the nucleus in the cytoplasm, and the ribosome are actually in the endoplasmic reticulum. Let me see. Yeah. All right, a very short kind of like slide about amino acids, because this knowledge is important to be able to define protein structure from a density derived by cryo-EM. As I said, there are 20 amino acids in a protein, and they differ by their properties. They are charged, positively, negatively, polar, and non-polar. One amino acid is actually quite an interesting molecule. It has an amino group, a carboxyl group, and then a side chain. And this side chain actually specific to each amino acid and define its property. During the formation of the polypeptide chain, you have actually a condensation event from point of view of organic chemistry, because we have the one water molecule is released when this polypeptide chain is formed. and it has a very specific organization geometrically, which helps model this protein when we don't have sufficient direct structural information. The way this has been done has been established by X-ray crystallography, and the first resolved structure was of hemoglobin. Here is the unique PDB coordinates of it, and this was done in the 1960s by Max Perutz. I cannot see the name here. I omitted the name. I will add this. Max Perutz gets the Nobel Prize for Chemistry in 1961 for resolving the structure of hemoglobin by X-ray crystallography with John Kendrick, and he's also the founder and chair and the first chair of the Medical Research Council Laboratory of Molecular Biology in Cambridge, who is actually pioneering the Cryo-EM methodology as a direct structure method. With this slide, I also want to introduce the protein data bank as a resource to find the structure for each protein. Basically, the protein structure, it's a text file which you can edit, which has the three-dimensional coordinate of each atom in the molecule of a protein, like the hemoglobin. Now, hemoglobin is an interesting molecule because it's a homotetramer, and it's kind of been resolved to high resolution, but all three direct structural methods. This slide is for the PDB 101 class for protein structure, and it's worth visiting because they have fantastic videos and teaching materials and introduction. The protein data bank was further managed by the Research Collaborative for Structural Bioinformatics, or the RCSB, which was established in 1971. And you can see the whole history here. To date, from 1961, when the first crystallography structure was resolved, we have 150,000 structure resolved. The first animal structure was deposited in 1989, and the first Cryo-EM structure in 1991. Now, I will very shortly go through the structure of the protein, how from a polypeptide or amino acid sequence you get to a quaternary structure which defines the function of each protein. The primary is simply the linear sequence of amino acids in these polypeptide chains. The secondary structure is a fixed arrangement of this amino acid, and it's quite a solid arrangement, very easy to visualize by each of the direct structural methods, and it's stabilized usually by hydrogen bonds. In the hemoglobin, we just have alpha helices, the other are beta strand. Unfortunately, the random turns is not really a preserved secondary structure, but is the third component of the protein, is predominant, especially in the proteins which essential function in the human body. The secondary structural components of a protein can be very well characterized by circular dichroism, and it's a good method to have complementary to any direct structural method. The tertiary structure is the folding of one polypeptide chains, and it's usually stabilized by weak interaction, mainly hydrophobic interaction. And the quaternary structure is usually several polypeptide chain, and that really define the function of the protein. And we all know hemoglobin is quite important for oxygen, and to carry oxygen in our body. Now, very shortly about the methods for protein structure determination. There are three methods to define protein structure directly, the X-ray crystallography being the oldest of them. And I have here illustrated all three methods with hemoglobin. And this is a 64 kilodalton protein. You can see the atom counts and so on and so forth. You can get all this information if you go to the PDB website and just type the code indicated under the images. In the X-ray crystallography, these three methods kind of have different potential from point of view of resolution, of point of view how the samples are prepared. One of the highest resolution of hemoglobin by X-ray crystallography is 125 angstroms. NMR is kind of gives multiple, it gives the flexibility, characterize the flexibility of the molecule, and it was one of the best structure is illustrated by these PDB coordinates. You can, I think they have like 20 kind of overlapping quaternary structures. And single particle Cryo-EM published a quite nice, they claim 2.8 angstrom resolution in 2018, and this is from a 200 kV microscope. All these structure, as I said, are deposited in the protein data bank, and you can access them. They are free for access, and you can actually observe all these images. Now, the benefits and drawback of all these methods have benefits and drawback. The X-ray crystallography, basically, you do need to have quite well-ordered three-dimensional crystals, which is not exactly a physiological environment or a native environment, and you do not record images. You do have diffraction pattern, and one of Max Parrot's achievement is they did this phase definition. You have to define the phase, because in an electron diffractogram you have only the amplitudes or the square of the amplitudes, which are intensity. In the NMR spectroscopy, you do need to have hydrogen atoms which are replaced with deuterium or other isotopes to be able to record the peaks. And in Cryo-EM, you do need to have very well purified samples to be able to record Cryo-EM images, and you have to have the technology to have multiple images, like hundreds of thousands of particles, to be able to obtain below 3 angstrom resolution. Cryo-EM, it's also the only method which actually give us the ability to observe or to study the structure of proteins in their closest to physiological environment, which is water. Now, one more interesting thing about this kind of holy tree, as I call them, in the structural biology, is how do they reflect compared to the size of the macromolecule. And hemoglobin is again a very interesting protein for this purpose, because 64 kilodalton or 60 kilodalton is an ideal size for X-ray crystallography. It's a big size. It's too big for NMR spectroscopy, and the paper which corresponds to these PDB coordinates explain that. Actually, it's one of the first molecules of this size they could resolve by NMR, and they did apply quite interesting technology. And for single particle Cryo-EM, it's way too small. Actually, until 2018, it was almost impossible to define its structure by Cryo-EM. Therefore, this size of macromolecules can unify all these 3 structural methods and also combine them, the information for the 3 of them. Now, how Cryo-EM relates to macromolecular structure. It was defined in 1968 by David de Rouzier, who is still Professor Emeritus at Brandeis, I believe, and Aaron Klug, who was recently deceased, who was one of the director of the MRC LMB founded by Max Parrot. And it is defined, is actually Cryo-EM was defined first and utmost as a structural method, which is based on electron scattering, not X-ray scattering, and the concept of 3D density maps and Fourier mathematics. Therefore, we do calculate 3D density map, and we do use Fourier mathematics to do that. And at the time, and still now, we have 3 main methods of Cryo-EM, how Cryo-EM is applied to solve protein structure. The first one historically is electron crystallography, and this is a paper from Baumeister and Stevens of 2000, and the numbers in gray reflect the resolution we have in the year 2000, and the numbers in red reflect the resolution we had last year, I believe, but it's quite accurate today, because it depends if you take the highest resolution or the average one. Therefore, electron crystallography, it's been derived for X-ray crystallography. The difference is that we use 2-dimensional crystal. We don't use 3-dimensional crystal. We also have images not only diffractograms. And in this way, we can combine the images with the diffractograms, because the diffractograms give us higher amplitude at high resolution, whereas the images will be truncated to about 7 angstroms. Now, the caveat with electron microscopy is that to obtain a full three-dimensional information for a molecule, you have to tilt it in the microscope, and you cannot tilt at 90 degrees. Therefore, you have always a missing cone, which is at least 60 degrees of the three-dimensional space, which we have to fill mathematically, therefore is not entirely accurate. The second approach is single particle analysis, and this really is the approach, the main Cryo-EM application in the modern days. In the last 10 years, it's kind of taken off tremendously, and it avoids the caveat of the missing cone, because the theory is that we have many molecules which are oriented differently in the image plane of the microscope. Therefore, the images in an electron microscope, there are projections, then we have many projections from 2D projection from a macromolecule which are oriented differently. What we have to figure out is how to build them back in a 3D structure. And this is done by the many software we have for single particle reconstruction. And the final, the least but not last, is the electron tomography. When it's like any tomography, you don't have multiple 2D projection. You have 2D projection of a single object. You have one molecule, you tilt in the whole three-dimensional space, and then you reconstruct. Recently, we have the single particle tomography, which, like the single particle analysis, average single molecules, just not 2D projection. It actually averages the 3D volumes, which are cut off electron tomogram. Again, the problem with electron tomography is the missing cone. Now, it's important to know the projection theorem, because if you have a symmetry, this will reduce significantly the number of tilts we need to have a full 3D reconstruction. Now, on the next slide, it's from electron crystallography. It's just one slide. It's my first work in Cryo-EM. It's where I started. It's what I did. We have a publication on that. I don't think the Cryo-EM map was not published, as the project was basically on electron tomograms, and it's on 2D crystals obtained by the lipid layer technique. This is a 2D crystals, a Cryo Electron micrograph of a cholera toxin B subunit, two-dimensional crystal. Here is the projection map. It's quite easy to get the projection map. It's straightforward when you know the symmetry and the corresponding electron diffractogram. You can see the ring, and 3.6 angstrom is the amorphous ice ring, which is the ice holding the biological macromolecules in the electron microscope. What is interesting, this was a 120 kV microscope. It was a tungsten filament. Actually, the Hitachi we have at CNS, it's a better quality than that. Therefore, from point of view of electron diffraction, you might not need the super performant microscope to date. And electron crystallography nowadays has been evolved as micro-ED. I don't believe they acquire images in micro-ED. They just do the electron diffractogram and then they extend the phase. It's like they do in X-ray crystallography. Now, the resolution revolution is what happened to Cryo-EM in the year 2015, 2016. And today, this is my favorite slide showing how we reached near atomic resolution for biological macromolecules by single particle Cryo-EM. Now, there are two barriers, and you can read all about this in this paper, which I still believe illustrate best what happened in 2015. And there were two barriers which we endeavor to break. This is the 2 angstrom resolution threshold and resolve structure of proteins below the size of 100 kilodalton. Both of these are still relevant today. Actually, 2 weeks ago, we had some breakthrough in the first a real breakthrough, which actually is producing now numerous, very fast sub-2 structure for many proteins. After 2016, after this slide, what was really developed was the membrane protein structure. We have numerous membrane proteins now resolved into 2 to 3 angstrom. Not all coordinates are deposited because most of them are done by companies such as Novartis and Lilly and Johnson and Genentech, I think in San Francisco. And also, what we achieved is 1.2 angstrom resolution for soluble protein, which are below 300 kilodalton size. 300 kilodalton above, it's quite easy to do single particle if you have a good purification. I have a couple more slides to illustrate the resolution revolution. And here we can see that the term was actually founded in 2014, and the growth is exponential. The map, as of May 2020, we have 11,000 Cryo-EM maps which are deposited, and here you have the website when you can search for the information. Unfortunately, these 11,000 maps are roughly half of them are unique structure. Most of them are repetitive. Now, the Cryo-EM map biotechnique is interesting because the single particle method totally overpowered all the other methods, and this has been progressive over the last 3 years. The single particle is now almost 80%, and tomography, single particle tomography, helical reconstruction, and crystallography has shrinked to almost 20% combined. What is more interesting is to see how the release of map per year follows the trend of the cumulative Cryo-EM map release. It's not exactly exponential, which means that we have like stepwise breakthrough of the resolution barrier. And if you follow the Cryo-EM map release by year and resolution, you can see actually the resolution revolution is happening in the 3 to 5 angstrom resolution range, mainly the 3 to 4 angstrom resolution range. This is the red bars. Now, how did it get so good so fast? It was, I mean, we all knew it, the people who worked in this field, but from outside, everybody said, wow, what really happened? How did you start solving like near atomic structure so fast at once? Well, it was a tedious process, and the Cryo-EM workflow can be kind of split in 3 main parts. One is the sample. The second is the hardware, which includes the electron microscope and detector, and the third is, of course, the software and the computing part. Now, the sample, it's the bottleneck of the whole process, and there have been tremendous advancement in that from point of view of biochemistry, the purification being of the most importance, grid optimization and preparation, the flash freezing and transfer optimization has been really has been polished tremendously over the last few years, and have contributed to this increase in high resolution structure. The biggest advancement, though, which really contributed to this resolution revolution, was the advancement in the hardware, the electron microscope and the detector. This is from point of view of guns. We have better and brighter guns like field emission X-FAG, which are brighter than the X-FAG, the one we have cold FAG, which are even brighter and have a better energy spread, better lenses, CS corrector, monochromators, faceplate, and so on. We have autoloaders, which stabilize the microscope and the samples in the microscope and allow loading of multiple samples, therefore collecting better and more data. And most of all, the direct electron detectors and energy filters coupled with these detectors. From a software point of view, there was also a revolution, mainly because our computers got better, using of GPUs, using of big clusters, know the clouds. we had some very new and very powerful imaging processing software like Reliant. Structural optimization and validation is probably will be in the next step. It has not changed much over the last 10 to 20 years. And all this resolution revolution led in 2017 for the Nobel Prize to the Nobel Prize for chemistry to Jacques Dubochet, Joachim Frank, and Richard Henderson. And you can see here illustrated in a structure how this reflects from 2013 to 2017. Strangely, this structure actually has not been produced from either of these Nobel laureates. It was produced by the lab of Sriram Subramanian in NIH in Bethesda. And I think it was the Cancer Institute, and the Zika virus was actually resolved at the time at 2.8 angstrom, I believe, by Michael Roseman's lab in Purdue. Here are the main paper from all three Nobel laureates describing their work if you are interested. And this is a timeline of how the single particle cryo-EM advanced to atomic resolution by Joachim Frank. And here, it of course didn't take only 5 years. It shows the main step. One was the introduction of field emission gun, and the second was the introduction of direct electron detectors. For imaging, now, Jacques Dubochet contributed mainly to the development of the sample preparation for imaging by the Cryo Electron Microscope, and to date, this paper in the Quaternary Review of Biophysics is the best describing what actually amorphous ice and cryo samples mean. You can see here the imaging of all 1, 2, 3, 4 state of ice and the corresponding diffractograms. Amorphous ice is the one you really want to see. And if you do, now we can do a fast Fourier transform on the software. We acquire the images. You can see if you have cubic or hexagonal ice in the direct images, and check this in Fourier space to make sure that your ice in your sample is good. Now, and there are a few challenges for biological sample in an electron microscope, and hence for cryo-EM sample preparation. All biological samples are up to 70% of water, and the nature if we dehydrate them or if you irradiate them. Therefore, we have to preserve these biomacromolecules or even subcellular structure or cellular structure in the high vacuum in the electron microscope. We have also to preserve them from radiation damage. Frozen hydrated samples are much more sensitive than negative stain samples, and we have to very acutely be aware of the electron doses we use to image this sample. And there is, and this depends actually of the voltage of the specification of your electron microscope. Higher is your voltage, higher can be your electron doses. We also have to, we endeavor to obtain better signal to noise, high contrast images. This is a challenge with biological samples because they have a very poor ability to scatter electron. They have low atomic mass atoms, and they're very comparable mass to the background, which is usually water. Therefore, getting the signal out of the noise is a challenge, is where the direct electron detectors come. And finally, to have a real structural atomic resolution, we do need to observe the weak phase in the object approximation. You cannot get an atomic structure for anything which is really thicker than 100, 200 nanometers. If it's kind of not very tightly packed, it's very weak scattered, you can get to 500 nanometers. Thicker it gets, lower is your resolution, higher is your radiation damage. Now, very shortly through, because negative stain is part of the cryo-EM workflow, because this is how we check our samples before we freeze them. And this is the same like for conventional TEM. It's slightly modified so it can match more precisely the Cryo-EM sample preparation methods, because we don't really look for images in the negative stain, we just use it more to evaluate our samples. And here is an example of the adeno-associated virus. Drawbacks and benefit of NS negative stain there, well, it's a classic slide because this is valid for all other application. Negative stain specimen is fast and easy to prepare. It is very cost efficient. It gives high contrast. It's more, the sample is more resistant to radiation damages, prevent radiation damage more, and is give you a very fast feedback for sample optimization. You can screen a lot of negative stain grids. For Cryo-EM, you spend much more time to go through your images and your sample. Disadvantages, it has a high background noise because the stain doesn't stain only your macromolecule, it stains also the solution, which is on top of your carbon film. You might have like structural artifacts due to dehydration. The resolution is of course low because of the grain of the negative stain, and you just don't visualize the real structure, you visualize the mold. And for the Cryo-EM sample preparation is also a very short slide. There are tons of literature on the net. You can go on YouTube and see different labs, how they prepare our samples. We have two machines for Cryo-EM sample preparation, one semi-automatic and one fully automatic. This picture is from the Katan semi-automatic plungers. Basically, you have to plunge very fast your sample, a very thin layer of your sample in liquid ethane, which is cooled down by liquid nitrogen. Hence, the technology we need has to be sophisticated. We use holy carbon film, which are deposited on an electron microscopy grid, which usually is a copper mesh, and hose varies from 1 to 2 micrometer in diameter. Here the samples usually has to be in the hose, and it has to be quite thin, and it has to be frozen quickly in the liquid ethane, so we avoid the formation of crystalline ice. I recently read they said 100,000 degrees per second. My recall from Dubouchet paper is 2000 degrees per millisecond. This is the rate at which your sample has to hit the liquid ethane, so you don't have crystalline ice. The machines now acquire sophisticated automatic cryo plunger. We have the VitruBot. It's very easy to use, and you probably don't have to worry so much about speed plotting and so on. Once you set the condition, your results will be quite reproducible. Now, the drawback and benefits of Cryo-EM sample preparation. The advantages of course, your macromolecules are preserved, fully hydrated in their native state, cooling down to liquid nitrogen temperature, stabilize your sample, therefore it prevents radiation damage. And of course, it allows near atomic resolution imaging because the resolving power of electron microscopes goes much beyond the size of the atom. Therefore, if you set up everything properly, you can see beyond the atoms, which is a case in material sciences. Disadvantages, it's radiation damage, is the very low signal to noise, requires much more time and effort to get images and structures that you would do with negative stain, and is quite expensive. It is very expensive. It's not easily accessible. Now, about a biological TEM, they are not exactly identical to the material science TEM because they have a few requirements which are not necessary for material sciences. They are though based on material sciences, and we benefit in Cryo-EM from the advances in material sciences. What is important to know is that when we really need to analyze is the phase contrast, which we cannot because you don't see anything in phase contrast. You have to introduce amplitude contrast, and we do this by defocusing. Now, radiation damage is quite important, and the type of radiation damage which actually affects biological samples are the secondary and tertiary radiation damage. This is why when you have higher acceleration voltage, you have lower radiation damage, because the time which the electrons spent in the sample are less, therefore you have less time to create free radicals and build up of gas. I mean, a better explanation is in the lectures of Professor Bell, which are fantastic regarding the all the specification of the electron microscopes, and especially the spherical aberration correctors. Now, one modern biological microscope has 3 cameras, should have, or best to have, one live camera which actually gets images of your screen. We don't have binoculars anyway, even if some microscopes have them, just like, I don't know, as an illustration of what was 10 years ago. We do have a CCD camera, because CCD camera give much higher contrast images, and you have the direct electron detectors which actually acquire the real data for structural analysis. Therefore, GAN is the main contributor to high resolution structure determination. CS corrector is the second contribution, and the direct electron detector is the third and most important. A little bit about the direct electron detector. They got established in the year 2013, and these 2 images are from a publication for data that were published in 2013. Basically, the best specification of a direct electron detector is that we detect directly electron. We don't have the information to go through a fiber optics, be converted from an electron to photon and back to an electron. The second advantage of a direct electron detector is that it has a very fast refreshing rate or frame rate. Therefore, we can record movies, which leads to an intrinsic dose fractionation. We can choose frames which are less irradiated, and we also can correct for motion of the macromolecules due to this radiation damage and like movement of the water molecule in the amorphous ice. Now, the direct electron detector, or any actually image detector in an electron microscope, is characterized by the detective quantum efficiency, DQE, and this depends on the physical Nyquist frequency, as you can see, and it's compared here to film. Nobody uses film anymore. You can see a CCD detector, the one we have here, it's in blue, and K2 Summit super resolution mode is in red. We have both of these detectors at CNS. This slide will give you a better understanding. It has more comparison here, but a better understanding how the Nyquist is characterized. This is basically a Fourier transform, a fast Fourier transform of an image. If you record your image, let's say at 5 angstrom per pixel, basically the highest resolution in the Fourier space, the highest frequency will correspond to 5 angstrom per pixel, and your Nyquist will be double of that. And this is your criteria of the maximal resolution you can obtain for given image. Why it doesn't move? Okay. All right. Now, this is the third part of the Cryo-EM workflow, which is the image processing, the data analysis and image processing. And I like very much this micrograph. This is the contribution mainly of Joachim Frank, the third Nobel laureate awarded in 2017. He worked more than 6 years on the ribosome, starting slowly to rise the resolution. In 2008, this was 22 angstrom ribosomes. They are huge macromolecular complexes, so almost 2 megadalton. And this resolution was raised to 3.2 angstrom 10 years, almost 10 years later. A standard classic image processing workflow starts first by the image evaluation, inverting, removing X-ray pixel, normalization, motion correction. And therefore, after that, we have to do a CTF correction, which is the focus estimation. I will not talk about CTF in this talk because it's a too big subject. Therefore, hopefully, I will be able to talk about that in a later stage, or maybe post, online courses and tutorial. Once we evaluate our micrograph and correct for CTF, we have to select the particles. And this is done by different software on the images. Once these particles are selected, we do calculate, we do classify this analysis by maximum likelihood methods in different classes. Basically, we match everybody with everybody, and we classify in classes for the particles which are most similar to each other. If it's a template-free classification, basically the criteria by which how many classes we will have, criteria by which the particles will be classified is we impose the number of classes we want to have, and we iterate this process until we have good class averages corresponding to the original particle or images of particles in our images. Once we classify the particles into the class averages, we have to build a 3D model. Usually we need an initial model or a tissue series to start that. If we have a previous structure or homologous structure, this will be better. We run this 3D classification and 3D reconstruction few cycles to get the final 3D reconstruction. Once we have a final 3D reconstruction, we have to validate this structure by different methods and build a model. And this is illustrated in this paper by John Robinstein, who did this for the ribosome at 3.2 angstroms. Now, this is another slide explaining the projection matching algorithm. Now, once we get all our set of 2D images classified in different classes, we have a 3D model from which we get projection. These projections are matched to each particle in our original data set and then reconstructed back in a 3D structure. This 3D structure is sectioned back to projection. These go back in the data set until these two images converge. And this all process, the qualitative criteria for this whole process is the cross-correlation peak between the original image and the 2D projects for the 3D reconstruction. The number of projection we need, it's again defined by the 3D, by the projection theorem of how many projection you have, and it depends highly on the symmetry. For this reason, one of the benchmark and proteins that are used to test all of the 3 steps of the Cryo-EM workflow, sample preparation, hardware, data acquisition, and data analysis, it's Apoferritin, as Apoferritin has 24 identical subunits and has a octahedral symmetry. Therefore, we need very few images to reconstruct the full 3D volume of the molecule. Also, this molecule, it's relatively big, is 450 kilodalton, and also it's very stable. It has a stable structure, and of course, it has a X-ray structure. Therefore, we can create a very valid initial model and test all the 3 parts of our Cryo-EM workflow to see how well we prepare the samples, how well we collect the data, and how well we analyze the data. And if you go in the protein database and you search for Apoferritin, you will see all the papers published with this molecule. This specific paper is for Columbia from the lab of Joachim Frank, and they used Apoferritin to validate one of their Cryo-EM grid preparation method. They publish a 3-angstrom structure, I believe, and in the next slide, a 3-angstrom structure basically it shows the whole step of the data analysis. Here is the original image, Cryo-EM image. It's a zoom view showing how the 2D projection of frozen hydrated apoferritin molecules look. Here is the 3D reconstruction, and all the subunits are color-coded. It's an alpha helical protein. These are alpha helices. Here is the validation, the criteria by which the resolution is assessed for Cryo-EM map is called the Fourier shell correlation plot. It's basically a Fourier shell correlation between two 3D reconstruction from two halves of the initial data set. And at 0.143 Fourier shell correlation, we have the resolution of our model, and in this case it's 3.04 angstrom. 0.4 is of course meaningless, therefore 3 angstrom is the resolution we can accept for this structure, and the model is also validated by fitting the atomic structure in the density derived by electron microscopy. Now, this was my favorite 3 apoferritin structures before what was published on 22nd of May. This is a 3 angstrom resolution structure by Joachim Frank. This is a 2.2 angstrom resolution structure by Scherslap in the MRC LMB in Cambridge, and this is a 4.7 angstrom resolution structure by Rousseau and Passmore in the LMB in Cambridge. Why I like them all? Because this one, the 4.7 angstrom structure, should be easy to obtain by anybody with little experience or known in Cryo-EM if you have the setup properly. As you can see, the information which is important to get these images is the type of microscope you have, and it's a 300 kV FAG, which is the must for near atomic resolution. It's a direct electron camera, K2 Summit or Falcon 2. These are the 2 which are mostly used. The pixel size, which define your physical Nyquist, and of course the dose. One of the things which also define the resolution of your structure is the number of particles which contribute to the reconstruction. The better is your imaging and your system, less number of particles you need to get the full reconstruction. And you can see that almost 500,000 were used for the 2.2 angstrom resolution, but only 3,000 were used for the 3 angstrom resolution, and only 6,000 for the 4.7 angstrom resolution. And you have, of course, the software used. I actually don't have the software for Rousseau and Passmore, but this must be reliant because it comes from the MRC LMB. And here, these are my last theoretical slide. I'm really very excited to show that because it came on the 22nd of May, and it actually visualized very nicely the water molecules in the apoferritin structures. Again, apoferritin, of course. And what it's really was discussed in the Cryo-EM community is actually, do we have electrostatic or electron density maps? What we are actually seeing, and why these density positive and negatives do not correspond exactly with the position of the atoms. Also, to get a 1.2 angstrom resolution structural apoferritin at this resolution to see the water molecule, it requires a very big team of people and serious modification to your hardware. And here it's really one of the small new revolution happening this year in the Cryo-EM field. And there are 2 papers. The second one is in the next slide, which actually tackle similarly the problem, tackle similarly and at the same time quite differently the problem. In the Shorshars paper at 1.2 angstrom resolution, it's with mouse apoferritin, and I do have to say that the one we use at CNS is the one we purchased from Sigma, and it was used in the slide with the 3 model I showed you. The one used for the 1.2 angstrom, below 2 angstrom resolution is it's expressed and purified properly, so you can really see the details. I'm not sure you can obtain this with a commercially buy specimen, but if anybody has access to this type of microscope, you're very welcome to try, and maybe we can prove we don't need so much purification to get resolution if we have enough good algorithm to select the good particles from the bad particles. Therefore, to get 1.2 angstrom resolution, what was really very important is the hardware improvement. It was introduced cold field emission electron guns. We use usually the Schottky FAG guns in Cryo-EM until now, which they have a very optimized energy spread of 0.3 electron volts. I believe the Schottky have 0.7, which improves the signal to noise ratio. And it's very important for resolution better than 2.5 angstrom. They used a very good, super performant Falcon 4 camera, which is coupled to a very special energy filter to additionally increase the signal to noise ratio. And below are the schematics from this paper illustrating that. They actually, what is interesting, at least to my understanding, is the way they obtained this structure. They actually corrected for spherical aberration computationally based on the energy filter specification. They did not use a CS corrector in their microscope. What they claim was quite interesting. They actually achieved a 1.2 angstrom resolution just from 400,000 particles, roughly 3,370 movies. They use, of course, the 300 kV cold FAG. They used the energy filter, and they said they collected all the data for 36 hours. I personally asked them, yeah, they did correction for the third and fourth order optical aberration, and they implemented correction for the curvature of the evil sphere. This actually was done before. In this paper, the main improvement is really the hardware. I did ask them personally how long it took them, what is the computer power, and how long it took them actually to calculate this structure. I haven't gotten an answer. But here is basically the raw images, the 2D projection, the 3D reconstruction. In microscopy, usually you don't just give one resolution, you give a resolution range. It's a dynamic resolution map because more flexible part of the molecule obviously will be calculated at lower resolution just because they don't have a unique conformation. Now, this improvement of this microscope compared here to their old reconstruction 1.64 angstrom resolution, which is acquired in the standard configuration. You have here in the HMS, in MIT, in Worcester, UMass Worcester, which is a Titan Cryo 300 kV XFAG microscope with a Gatan K2 Summit camera or Falcon 3 or Falcon 2. They will all give you probably for Apoferritin, well purified Apoferritin below 2 angstrom resolution. 1.2, it's however a significant improvement because you can see the electrostatic maps. You cannot see this at 1.65 angstrom resolution. Another achievement in this paper, they actually did a 2 angstrom resolution of a membrane protein, and this was also spectacular, but I'm not going to discuss this in this talk. The next paper appeared in the same journal on May 22nd, was from Göttingen, and they also used an improved microscope. Their microscope, however, had a spherical aberration corrector, BCOL, they call, and they have, of course, a cold FAG. They claim a very small energy spread. I mean, if you want to learn about CS correction and cold FAG, one of the world experts is actually Professor Bell, and this is very implemented at CNS. And I believe the new megascope, the Hitachi 300, which is installed at CNS, it's correspond or it's above the specification of this microscope. The only part it lacks for high resolution Cryo-EM is that it doesn't have a direct electron detector, but I really hope we will get one at some point. They also use the monochromator, and they of course correct up to fifth order aberration. What is interesting, there was only 4 people involved in this. For this structure is 1.25 angstrom, even if I'm not sure 0.5 angstrom is really how easy is to calculate this by the Fourier shell correlation. And they use like 1.5 million particles and more than 11, almost 11,000 movies. Therefore, if you are interested how far you can go in Cryo-EM, please do check these 2 papers. They are really outstanding. It's just amazing we could achieve that in this year. The cold fact, just to give a note to our Japanese colleagues, the cold fact was actually first used in this paper in 2019, and they had a structure of apoferritin at 1.54 angstrom. Actually, why do we go to this resolution? Why we are so desperate to go beyond 1.5 angstrom is because the CC bond in the polypeptide change is 1.54 angstrom actually. And we need this dimension to be able to exactly calculate the 3-dimensional organization of the atoms in a protein molecule. I hope I have 10 more minutes. I want to very fast introduce what we have at CNS. And this is a single particle cryo-EM analysis workflow, which is which I don't know if it's still there. Hopefully it is. Which kind of this figure is from Thermo Fisher. They are the main producers of the Cryo-EM microscope for high resolution structural analysis, and everybody who can afford buy them because they are really extremely efficient. What we have in CNS is much smaller, and what we can do from this workflow is the negative staining, the sample vitrification, the sample screening and data acquisition, and we can do a 3D reconstruction with moderate resolution. We are equipped to all that. And here it's our Cryo-EM microscope. It's a 200 kV SFAC, which means less bright, therefore we cannot go at much higher magnification because of the radiation damage, but we do have a GATAN K2 Summit direct electron camera, which is important. This is the box here on top, which actually control this camera. The camera is of course inside the microscope. What is interesting about this microscope is that this is a prototype. There are only 4 or 5 in the world. I know only of 2 others. One is in the Netherlands, in Maastricht, the other in Singapore. Nobody can tell me when are the other 2. Maybe they're not operational. It has of course an autoloader. It's connected to liquid nitrogen dewar, which means it's cooling constantly. You can use it remotely for 4 to 5 days without even touching it. We have a remote control room where we have here is a microscope user interface. Here the CCD camera monitor. This is the direct electron camera monitor. And here is our VitruBot, which we use for the sample preparation, negative staining, and this is a semi-automatic GATAN cryo plunger. We are reconstructing actually this area. Hopefully we'll do it shortly because we are opening on Monday. We have a training attached to this microscope. Even if you don't have a sample, you might still want to see how this technology works. But even if you don't do your sample, I would still suggest get the training so you know actually what you're doing and how it is done. There are very few microscopes which are available to for training in the structural cryo-EM because they're constantly used for collecting data. Therefore, many people come just to see how actually it's done. And because we have a prototype, we actually can go in every step in more detail than you would with the modern machines. Some of them are done like black boxes. You just go in, go out, but you don't know what's happening in the middle. Our machines will let you see what actually is happening in the middle. And therefore, we definitely can get images of any sample for like 2 days training. And we have, of course, complementary training, which will go through all the structural analysis and all the processes and steps we use to collect and analyze the data. Here is some illustration and what actually is achieved during the basic and intermediate courses. The basic is on negative stain because you just have to learn how to image. Because it's the SFAG, we cannot really image at high resolution, the 2.5 angstrom per pixel on the CCD camera. These all images are on a CCD camera. This means you won't achieve more than 5 angstrom resolution in cryo, because above that the radiation damage will be too strong, and you have to move on the direct electron camera when the contrast is too low. For the advanced Arctica Cryo-EM course, we use Apoferritin, and this is the one which came out of the bottle as sent from Sigma. This is horse spleen Apoferritin, and you can see you cannot actually zoom the direct electron camera images because you won't see anything more. These are the CCD images. And you also will need more concentrated sample. These samples are all the same concentration. Therefore, it has to be over-concentrated negative stain. This is what you're checking. If you have this concentration in negative stain, then yeah, you will get good images in cryo. You will be able to do some analysis. Now, here are some of the projects we work and compared the negative stain and cryo-EM images. These are liposome for drug delivery. What I forgot to mention is actually one of the goals of the intermediate Arctica training is to visualize the lipid bilayer. You won't be able to see this bilayer if your defocus is not right or if your radiation is too high. It will just melt away or it will not be visible because of overlapping frequencies. You also evaluate the images in cryo-EM in the Fourier space. You have to see good tone rings, and they should be spherical and they should extend as further as the Nyquist resolution allows it. And of course, you're scattering material. Now, one of the projects we do, this liposome for drug delivery, this is simple imaging. Imaging in cryo-EM, it's much more complex than in negative stain because the information has to be recorded before it's visualized, and it has to be recorded properly. Therefore, you really have to practice on the standard specimen. The other thing is that the sample is not uniform. If you have a complex sample, you will have to go look over 300 squares. Each square has like 40 holes, and this will take tremendous time and kind of looking power. You have to look at each image over and over, and you have to know roughly, and you have to kind of translate the information back in your brain. Cryo-EM will really be applicable well for non-structural projects when we have deep learning software which will be able to analyze huge amount of data and make sense of them. I don't know how you teach a machine to look for something you don't know actually if it's there or not, or might be something different, but I believe we will get a solution in 10 years to that. Here are negative stained and cryo-EM micrograph of solubilized membrane channels that are actually with antibodies. If you look well, you will see the antibodies both in negative stain and in cryo-EM. Here are negative stain and cryo-EM of adeno-associated viruses that are used for gene delivery. As you can see, the images are drastically different. You really have to know how to look at them and to translate properly the information. These are exactly the same magnification, not exactly the same defocus. And two single particle projects we did at CNS. This is the RNA polymerase project. This was done with the CCD camera. It's like take a week. It took like 125 images, 30,000 particles, and you get 11 angstrom resolution without a problem. What you need is really a well-purified protein. The other project which was carried very far, but you don't know group in Dana-Farber Institute in PKU, is the 26S proteasome project. This was acquired with the direct electron camera in super resolution mode, which actually allowed to achieve reconstruction 2.8, 3.6 angstrom resolution. It's a very, very flexible macromolecule. Therefore, we did need to select a tremendous amount of, to acquire a tremendous amount of movie, almost 45, and select nearly 3 million particles, which was done, of course, with neuronal network and deep learning mechanism. You cannot do this by hand. And here are 2D projection calculated from the original Cryo Electron Micrograph. It's a quite big molecules, therefore it's allowed to do this high resolution with a 200 kV microscope. And when you have a flexible molecule, what you do multi-refinement. You basically mask one part which is flexible and you calculate the part which is rigid, and we alternate. In this way, we calculated 7 different conformation, and here is the movie of how this conformation was combined to create a dynamic model of substrate processing by the human 26S proteasome, and it was published last year in Nature. You can, of course, see the paper. There are more movies and much more explanation, but this is a tremendously difficult work, and it took over 2-3 years to achieve. It was a stepwise process, and data were combined to many Cryo-EM sessions. The first of one, I was not present, just at the one which we carried in 2017 and 2018. But a full Cryo-EM high resolution project will culminate in a structure like that. However, as the things stand right now, at the time, 2017, we didn't have except in Worcester, we didn't have high resolution Cryo-EM microscope in Boston. Now we have in HMS 2 microscope, in MIT 1 microscope. I'm not sure. Boston University is buying a 200 kV, and some companies have 200 kV. The nanoimaging service actually just built a company, Woburn, I believe. They have a couple of crises as well. Therefore, with the technology we have at CNS, what will be, if you want high resolution structure, what you should do really is characterize and screen your samples. And when you have good grids, which promise high resolution, these grids can be directly translated in a high performance Cryo Electron Microscope. There are also 3 centers in the US who will who will do data acquisition of grids which has been pre-screened with technology we have at CNS. Some other project I did a composites like RNA, origami, linear, superimposed lattice, and so on. These are nano ribbons. There are carbon nanoribbons. We have polymer nanoparticles, very hard to imagine because they are very, very small and have a very low signal to noise ratio. And of course, multilayer vesicles with peptide. These vesicles could not be imaged at higher than 4.4 angstrom per pixel because they're so big, they bow immediately when they see even one electron. Therefore, it was a challenging project, but we got very nice pictures. And finally, my final slide about the future of Cryo-EM, structural Cryo-EM, and hopefully what we hope to develop as well at CNS, and this is the cryo-electron tomography. The principle of electron tomography, it's quite simple. You have a macromolecule or a cell section, you tilt it in the microscope, and then you reconstruct back in a 3D volume. We use usually one tilt axis. Dual tilt axis is a bit hard to accomplish with the Cryo-EM, but this is the concept. And as I don't have a cryo-electron tomogram from the Arctica yet, I took a couple of movies here. This is from a paper, like this is how electron tomogram of a section through a cell looks. This type of cryo-electron tomography has to be coupled with cryo-TEM. I forgot that. with the Cryo-EM, with the Cryo-FIP, sorry, because the Cryo-FIP can generate small lamellars. Here is the Thermo Fisher movies, they're quite nice. They can generate small lamellars, which can then be mounted in Cryo-EM grid and loaded in a Cryo Electron microscope. Then now we have this Cryo-FIP, which Stefan is working with, and we hope then we will combine and set up the Cryo Electron tomography workflow on our Cryo-EM, hopefully by the end of this year. Depends on how the reopening and everything works. And the thing with cryotomography is that we cannot put whole cells. They have to be sectioned. It shouldn't, to get this resolution, you shouldn't really be above a few hundred of nanometers thickness of these sections.
You have a question in the chat, Svetla.
Yeah, I'm trying to, not trying to.
Would you comment more about the electron dose and the Nyquist fraction relationship?
Yes, absolutely. This is a whole lecture, which you are definitely more qualified than me to give, but I will definitely explain. I just wanted to see, I had a, I'm looking for a movie. I'm looking for YouTube. Yeah, I just want to show that because it's a lot of information there. This is from the diamond light source, which is pretty much the synchrotron in the UK, in Oxfordshire. It's a comparison here to the SLAC in Stanford, and it tells you how cryo-electron tomography works, and everybody agrees that this will be the next revolution in cryo-EM. But for the moment, we do need to get very thin cryolamels in cryo-FIP to be able to collect this quality of data. And what I wanted to see is that if you go to this YouTube link, you can load all this information. You have tons and tons and tons of cryo-EM presentation. I'm going now to have a look on the question.